Welcome, everyone. Thank you for joining us for tonight's webinar. My name is Maggie Howell, and I'm the Executive Director of the Wolf Conservation Center. Just a few items before we get started. If you have any questions during the presentation, please just type them into the Q&A box. You can find it on the control panel at the bottom of your computer, and we'll provide time for questions at the end of Morgan's talk. Also, a recorded version of this webinar will be available on our website at www.nywolf.org within a day or two. All right, so today we're joined by Morgan Bragg, who has generously offered her time to discuss the relationship between the gut biome and gastrointestinal health of captive red wolves. Morgan Bragg is a PhD student working in collaboration with George Mason University and the Smithsonian Conservation Biology Institute Center for Conservation Genomics and Center for Species Survival. Bragg's dissertation focuses on host microbiome environmental interactions and their influence on gut health, on host gut health in captive red wolves and maned wolves. She recently received her master's in 2019 from George Mason University, where she studied the relationship between gut bacteria, diet, and gastrointestinal health in red wolves. Well, that was a mouthful. I had a lot of problems there. I apologize. But I'm really excited to get started. So without any further ado, I will turn the time over now to Morgan. Hey, Morgan.
Hi, Maggie. Thank you so much for that great introduction.
Thanks.
Hi, everyone. So like Maggie said, I'm going to be talking about a piece of my master's research that focuses on the relationship between the gut microbiome and gastrointestinal health in the captive red wolf. And I did just want to give a very clear disclaimer before I started that these results are not published. I am in the process of writing up my manuscript to submit for publication, but please do just be aware that this is not published. And so thank you, Maggie, for that great introduction. But I wanted to give a little bit of an introduction about myself that's maybe like away from science. And so if I'm not doing grad school or science related things, you can catch me outside with my dog, Molly, which you can see here. Like Maggie said, I am a PhD student at George Mason University. I got my master's there as well. And I got my undergrad degree from University of Tennessee in Knoxville, and I'm a huge Vols fan. When I'm not doing grad school or science things, I also like to tend to my indoor and outdoor plants. I have a little garden going on my balcony. When it's non-pandemic times, I do enjoy to travel as much as I can. And so these are some photos from when I went to Costa Rica back in January. And yes, this is a little sloth down here. I enjoy getting out on the water and kayaking whenever I have the time. And I am also a bit of a foodie. And so I'm actually going to turn my video off for the rest of the presentation, and then I will come back for the Q&A session. All right, so let's talk about red wolves now. So the American red wolf is a medium-sized canid that is smaller than a gray wolf, but bigger than a coyote. In the wild, red wolves live in packs. They're multi-generational packs of about 5 to 8 individuals, and it depends on varying environmental factors. The defining reddish color can be seen behind the ears, down the legs, and on the muzzle of the red wolf. This species is monogamous, meaning that they breed with the same mate for life unless something happens to their mate. The red wolf is highly elusive, very rarely being seen by humans. And so I wanted to share an experience that I had back in December of 2018 when I had a chance to go to Alligator River National Wildlife Refuge for a week. And so in this refuge is where the one reintroduced population of red wolves lives. And so I got to go to the refuge for a whole week, and I got to spend my time with U.S. Fish and Wildlife, hanging out with them, riding around in the truck, asking them questions, and helping out with their yearly trapping efforts. And while I was there for that week, on the last day, we actually caught a red wolf. And so you guys can't see it in this bottom photo here, but this red arrow is pointing to a red wolf. And so, of course, I was very excited to be able to see a real live red wolf with my own eyes. And I was also very excited because I got to collect 2 fecal samples from the wild population to include in my study. And then I have some landscape photos up here at the top for you guys to look at. It was really cool as a grad student to be reading about this area for about a year and a half, and then to actually go out there and see it with my own eyes. The refuge is made up of a lot of agriculture lands. And so it's these long strips of agriculture land that's divided up by these drainage ditches because they have to pump water out in order to produce any agriculture crops. So it's a very beautiful place. It's quite flat, but very beautiful. And then on this bottom right-hand corner, I grabbed a little picture of this nice paw print that we saw when we were walking around. And I did get to see some other wildlife too. So in the bottom left-hand corner, we have this nice big black bear. So you're probably wondering, why should we care about red wolves? We should care about red wolves because they are our only native wolf to the United States. We should take pride in the fact that the red wolf is living only in our country. I wish that people had the same sense of admiration towards the red wolf as we do towards the bald eagle. We should be really excited and we should support the species and we should celebrate the red wolf because, like I said, it only exists here in the United States. As an apex predator, the red wolf plays an important role in prey species population management, typically taking out the weak and the sick. And I think the thing that draws me to the red wolf story the most is the fact that they're just such a misunderstood species, and I feel like they didn't really have a chance from the get-go. Predator control programs and private citizens have decimated red wolf populations throughout the 20th century. So much so that in 1980, there were only 14 true red wolves that were left in this small area in Texas and Louisiana. U.S. Fish and Wildlife went in, removed those last 14 red wolves, and they started the breeding program. And once that was done, the species was declared extinct in the wild. And so a lot has happened since 1980 up until present day, too much for me to go into right now. But as of today, the red wolf is listed as critically endangered by the IUCN. There are approximately 275 wolves that live in the Species Survival Plan, or SSP, captive population, with approximately 20 wolves living in Alligator River in the reintroduced population. This species heavily relies on its captive population for continued existence. And as vital as this SSP population is, captive management can be challenging, and individuals can display health disorders that are not described in the wild population. An example of this is gastrointestinal disease in the captive red wolf. And so from 1992 until 2012, 21% of adult captive red wolf death was directly linked to gastrointestinal or GI disease, with inflammatory bowel disease or IBD specifically killing 8 wolves. And IBD is a type of gastrointestinal disease. An additional 25% of wolves that died from 1992 until 2012 had non-lethal GI lesions, with IBD specifically found in 22 of those wolves with the non-lethal lesions. And from 2018 until 2019, we did have 4 individuals that died in the SSP population from gastrointestinal issues. And so IBD is a group of diseases that's characterized by the inflammation in the small or large intestinal mucosa. So if we want to refer to this visual on the right-hand side, on the top, we have a histology slide of a normal duodenal mucosa. And as you guys can see, it has, nice defined ridges here. We don't see any red, inflamed looking areas. Compared to the bottom, we have a histology slide of inflamed duodenal mucosa. You can see that these ridges are much less defined here, and we have a lot of red, inflamed looking cells. IBD affects humans, canids, rodents, and felids. And when I talk about canids in general, I'm just talking about, domestic dogs, wolf species, coyotes, foxes, etc. And some symptoms of IBD in canids include chronic vomiting and diarrhea. Inflammation along the mucosa makes it hard for the intestines to absorb nutrients. Thus, malnutrition is common in individuals who suffer from IBD. The etiology or the cause of IBD is unknown. However, there's evidence that complex interactions between GI microbiota, which is the bacteria in your gut, environmental factors like diet, and genetic predisposition play a role in causing IBD. And so the gut is filled with millions of bacteria, fungi, archaea, and viruses. However, today I am just focused on the bacteria in the gut. And we actually get a lot of benefits from this bacteria in our gut. And some of the benefits are that they protect against pathogens, they aid in immune system development by learning which bacteria are commensal or beneficial for us, and which are pathogenic, which are not so beneficial for us. gut bacteria can ferment non-digestible dietary carbohydrates and produce a byproduct called short-chain fatty acids, or SCFAs. And these SCFAs, specifically butyrate, propionate, and acetate, are really important for immune cell movement, function, and production. And lastly, gut bacteria can synthesize vitamins B and K. And vitamin B is important for carbohydrate and fat digestion, and vitamin K is important for proper blood clotting. So there's no literature out there right now that specifically addresses the gut bacteria community in the red wolf. Therefore, we have to lean to domestic dog literature to begin to understand the canid gut. And so some common bacteria that are in the domestic dog gut are firmicutes, which mainly produce the SCFA butyrate. And this bacteria has been associated with a high carbohydrate diet in humans and dogs. We have Bacteroides, which produces the SCFAs acetate and propionate. Proteobacteria, which is a diverse type of bacteria. It contains many commensal bacteria, and it also contains some pathogenic bacteria. We have actinobacteria, which is important for the maintenance of the gut barrier homeostasis. And lastly, fusobacteria, which is a commensal gut bacteria that colonizes mucous membranes and has been associated with a high protein diet in domestic dogs. And so I'm going to be using a term, the gut microbiome, throughout my talk. And when I talk about the gut microbiome, I'm talking about this entire community of bacteria that's in the gut of these individuals. It has been frequently documented that the domestic dog suffering from IBD will have shifts or changes in their indigenous gut microbiome. And so on the left-hand side, we have the healthy gut microbiome of a domestic dog. It contains the same 5 bacterial phyla that I just spoke about. And then on the right-hand side, we have the gut microbiome of a domestic dog suffering from IBD. It contains all the same bacterial phyla, however, the relative abundance of these bacteria has changed. And when I talk about relative abundance, I'm talking about the proportion of each type of bacteria, so their ratios. So if the bacteria is green, it means that there was an increase in the relative abundance. And if the bacteria is red, it means that there is a documented decrease in the relative abundance in the gut microbiome of a domestic dog suffering from IBD. Firmicutes is both green and red because there are mixed results in the literature. So this shift in the gut microbiome can be caused by a whole slew of things. It could be lifestyle modifications, diet, antibiotics, host genetics, stress, or infection. However, today, I'm just going to be focusing on how diet impacts the gut microbiome. Although the domestic dog and the red wolf are closely related species, there are still evolutionary differences that separate the two. There is evidence that domestication of the dog from the wolf was marked by an increased ability to digest a starch-rich diet. A paper put out in Nature by Axelson et al. Assessed expression and activity in genes and enzymes that are involved in starch digestion. One of the genes that they looked at was the AMY2B gene. This gene encodes for amylase expression in the pancreas, which allows the domestic dog to digest starch. Humans have amylase activity too, we just have it in our saliva. And so from this paper, we have this figure here, and on the x-axis, we have the copy number of that AMY2B gene. And then on the y-axis, we have the number of individuals that possess that respective copy number of the gene. The blue indicates the wolf, and the red indicates the domestic dog. Interestingly, all wolves only had 2 copy numbers of the AMY2B gene, compared to the domestic dog had a varied number of copies from 4 up to 30. Authors reported an average 7.4-fold increase in the copy numbers of the AMY2B gene in the domestic dog compared to the wolf. So based off this evidence and literature, we see that the wolf is not as adapted to this starch-rich diet as its cousin, the domestic dog. Because of this, the approach to diet may need to be reconsidered so that the daily diet of the red wolf does include whole meat. And so based off literature, we know that red wolves in the wild eat white-tailed deer, raccoons, rabbits, rodents, and other small mammals. Whereas in captivity, the wolves that were involved in my study had one of 3 different diet types. They could eat kibble, which is a meat-based dry dog food that's approved for the domestic dog. This is the most common diet type in my study, with 35 individuals eating a kibble diet. And while this dry dog kibble is nutritionally balanced, it does contain a lot of starch, with 30 to 60% of the dry matter being carbohydrates. They could have a mixed diet, which is a mix of kibble and commercial meat. And this commercial meat is typically like a log meat that is mostly comprised of horse meat. So I have 10 individuals in my study that eat a mixed diet. And then whole meat or carcass. I have 3 individuals that eat this diet, and it consists of white-tailed deer, elk, wild turkey, beaver, rats, or chicken. And as much as facilities do try to mimic the wild diet in captivity, these captive diets can be far from the nutritional composition that a wild wolf would get in its natural environment. And although we know that this kibble diet may not be the best for the red wolf, the Association of Zoos and Aquariums, or AZA, is trying their best to provide the highest level of care to the red wolf and all captive animals. Like I said before, captive management is really, really, really hard. And red wolf diet and actual large canid diet overall is a topic of high interest for AZA. However, it can be difficult to change AZA diets because understandably, they need to be provided with a lot of sound science to consider or enact any diet change. And so that's exactly why I conducted the study that I did, to provide science. And so that leads me to my main overarching question, which is just how do red wolf gut microbiomes differ among diet types? So for my sample collection, I collected fecal samples from 50 individuals. 48 of those individuals were from captive facilities, from 8 different captive facilities. And then I had the 2 samples from the wild population in Alligator River. And so I have this table here on the right-hand side, and it shows each facility that participated in my study. They're also denoted by these black dots on the map, so you can see their spatial relation. It includes the number of fecal samples that were donated by each facility, the number of wolves that participated from each facility, and the diet type of each facility. Typically, each facility feeds the same diet to all wolves, except for Point Defiance Zoo and Aquarium. 26 of the individuals that were involved ate a kibble diet, whereas 2 individuals, there were 2 females, they were on a whole meat diet because they had been diagnosed with IBD. So for a brief overview of my methods, I had these facilities graciously ship me these frozen fecals, and then I get the fecal samples, I defrost them, and then I take a little piece of each fecal sample, and I use a QIAGEN kit to extract DNA from each individual fecal sample. I then take that extracted DNA and I do a 2-step polymerase chain reaction, or PCR, using this machine here. During this 2-step PCR process, I target the 16S rRNA gene. And I target this gene because it is a highly conserved region within the bacterial genome that allows us to identify bacteria. And so I then take that PCR product and I place it onto a sequencer, which is this machine here. The sequencer works its magic and it takes that PCR product and it converts it into individual DNA sequences. I can then take those DNA sequences and plug them into a computer program that allows me to assign taxonomy to each individual DNA sequence. So that's how I can identify which bacteria types are present in each gut microbiome. And this methodology of targeting the 16S rRNA gene allows me to get a broad scale overview of the gut microbiome composition in the red wolf. And so once I had all of my sequence data, I had to do a bit of cleaning up before I could do any sort of analyses. And so first, I filtered out any poor quality sequences. I then removed any contaminant taxa bacteria using my negative controls. And then I'm not sure if anyone noticed, but on my sample collection table, I did have some wolves that donated more than 1 fecal sample. However, I only needed to include 1 for analyses. So I used a random number generator to just choose which sample to include from those individuals. And after I did all of that, I was left with 443 bacteria in 49 samples. The top bacteria that were found in the gut of a red wolf are firmicutes, Bacteroides, Fusobacteria, Proteobacteria, and Actinobacteria, which are the same top 5 bacteria that are found in the gut of the domestic dog. So I assessed the gut microbiome at 3 different levels. So the first level I looked at was alpha diversity. And with alpha diversity, I'm concerned about within community variation. And so if we want to refer to this visual here on the right-hand side, we have these communities that are outlined by these orange dashed lines. And so when I am interested in alpha diversity, I'm only concerned with the variation within this one community. And to put this in the context of my study, each of these communities that are outlined by the orange lines represent a gut microbiome of an individual. So this is a gut microbiome of an individual, this is a gut microbiome, and etc. And then these trees that are inside the boundary represent the bacteria that are present in the gut. So the way that I assessed alpha diversity was by looking at bacterial taxa richness, which just asks how many different types of bacteria are present. The next level that I looked at was beta diversity, which is between community variation. So again, if we want to refer to the right-hand side with this visual, when I'm looking at beta diversity, I'm concerned with comparing the variation from this community to this community, or comparing the variation of this community to this community, and so forth. So to do that, I examined unweighted unifrac distances. And that looks at relative abundance of each bacteria present while also considering their genetic relationships to one another. And lastly, I looked at changes in the relative abundance at the phylum and taxa level. And when I say the phylum level, I'm talking about a very general high order level. So it's just a very general overview. Compared to a taxa level, you guys can think of it as species level. So it's very fine detail. So to look at these changes, I did differential abundance analysis and indicator species analysis. So the results from alpha diversity. We have this box plot here, and on the x-axis is our diet types. And on the y-axis, we have our bacterial taxa richness. And so we found that the gut microbiome of the wild diet had significantly lower bacterial taxa richness than the mixed or the kibble diets. And so that means that the gut microbiome of the wild diet has lower number of different bacteria present compared to the mixed or the kibble diet. And although facilities do try to be as hands-off with the red wolves as possible, contact is inevitable, whether it's direct through maybe having to do a health exam or give a vaccination, or indirect through just feeding, giving enrichment, or things similar to that. And this contact between humans and wolves in a captive setting could be accounting for this higher bacterial richness in the gut of these captive wolves. Studies have shown that it is possible for bacteria to transfer from humans to animals when they are living close by. Now moving into beta diversity results. So overall, we did find that gut bacteria community composition did differ among diet types. And so this plot here in the middle is called a PCOA plot. And to interpret this plot, each dot is the gut microbiome, the overall gut microbiome composition of an individual wolf. So this represents a wolf, this represents a wolf, this represents a wolf, and so forth. If the dots are close to each other, it means that their overall gut microbiome composition is similar, versus if they are further apart, it means that their gut composition is different. Each color represents a different diet type, and then these circles are just 95% confidence ellipses for that respective diet type. We found that diet explained approximately 16% of the variation seen within the data set, which is fairly good for a microbial ecology study. We found that specifically the differences lied between the kibble and all other diet types. So that means that the overall gut composition of the kibble diet type was significantly different than the overall gut composition of the mixed, whole meat, and wild diet type. However, I did also find that dispersion was significant. And when I'm talking about dispersion, I'm talking about the amount of variance within the data of each diet category. So because dispersion said it was told me it was significant, that means that the variations of the data within these diet categories are not equal. And so sometimes when like you're trying to compare these different diet types and they have unequal variances, that unequal variance can be what's driving the significant difference that we're seeing in beta diversity. So we just have to be kind of careful with interpreting this result, especially because I do have unequal sample size with the whole meat and wild diet. So again, just generally being cautious with interpreting this result, but overall we did find that gut bacteria community composition did differ among diet types. And now looking at changes in relative abundance at the phylum level, which again is that high general level. And so we have this relative abundance plot here. The x-axis is diet type, and again, N is the number of individuals in that category. And then on the y-axis, we have the relative abundance or the proportion. So with the kibble and the mixed diet, we see an increase in the relative abundance of firmicutes, which is this pink color, in comparison to the whole meat diet type. And so again, firmicutes is that phylum that is associated with the diet high in carbohydrates in the domestic dog. And like I said before, the kibble is made up of a lot of carbohydrates. So it's possible that this kibble diet is leading to this increase in the relative abundance of firmicutes. I did though find it quite interesting that the wild wolves have this high relative abundance of firmicutes. But of course they're getting this from a different food source in their natural landscape. And then with the whole meat diet type, we saw that there was an increase in the relative abundance of fusobacteria compared to all other diet types. And fusobacteria is that phylum that has been found in the gut of other marine carnivores and the domestic dog, and it has been associated with a high protein diet. So again, I'm not super surprised to see an increase in the relative abundance of fusobacteria in the whole meat diet, which is largely just protein. And then lastly, we saw an increase in the relative abundance of bacteroides, which is gray, in the captive wolves compared to the wild wolf. And so this bacteroides phylum is the one that produces the SCFAs, acetate, and propionate. And then changes in relative abundance at the taxa level, which again, you can think of it as species level. So this is a heat map. And so each color or sliver of color represents a wolf. If the colors are darker, it means that there is a higher relative abundance, versus if they are lighter, it means that there is a lower relative abundance. And so we found that there were 3 bacterial taxa associated in high relative abundance with diets that included kibble. And after painstakingly searching the internet for what these bacteria do, I found a little bit of information about them. So the first one is Cantenibacterium mitsokai. This bacteria has been related to carbohydrate fermentation and propionate production in felids, and it has been associated with high fat, high sugar diets in humans. Next, we have Prevotella species, which similarly has been related to carbohydrate fermentation and propionate production in felids and domestic dogs. This bacteria taxa has also been documented to have an increase that has been related to humans and mice with irritable bowel syndrome suffering from recurrent diarrhea. And then lastly, for the kibble associated ones, we have Holdemanella species. And so this species has, or this taxa has been linked to butyrate, acetate, and propionate production, and similarly has been linked to gut microbiome changes due to high fat, high sugar diets in humans. And then on the opposite end of the spectrum, we found 2 bacterial taxa that were only found in the wild wolves. And so the first one is the Clostridium 6 species. And so this taxa has been documented in the gut microbiome of healthy domestic dogs and cats. And this taxa has also been documented to prompt anti-inflammatory reactions in the gut of domestic dogs. And lastly, we have this Lactococcus species. This species has been identified in the gut of other canids, including Arctic wolves and coyotes. And certain strains of this taxa are used to cultivate probiotic dairy products for humans. And so it's possible that this non-natural captive diet is attracting non-natural bacteria or altering the gut microbiome, the native gut microbiome in order to aid in the digestion of this captive diet. However, these non-natural bacteria and/or the alterations in the gut microbiome can impact GI function. Diet can reinforce intestinal barrier structure and function via the production of these SCFAs. And these shifts in the gut microbiome caused by diet can impact the amount of SCFAs produced. This could be due to an alteration in the amount of SCFA producing bacteria, or it could be because of downregulation of G protein coupled receptors, or GPCRs. And so when these receptors are activated by SCFAs, it triggers a signaling cascade that regulates immune function. And I know that that can be kind of tricky to think about, so I have this visual here to help us kind of move through this idea. So we can imagine, right, we're eating our daily diet, it's going into our gut, it's being digested. Any of those non-digestible dietary carbohydrates are being fermented by the gut bacteria, and the byproducts of that fermentation are these SCFAs. And then these SCFAs can be taken down to the intestinal epithelial cells and diffused to go do various functions. And these intestinal epithelial cells are just the cells that line the surface of our intestines. This is also where those GPCR receptors live as well. The SCFAs and the GPCRs work together in order to maintain intestinal barrier integrity by inducing mucosal healing and suppressing inflammation. The intestinal barrier is essential in making sure that the inside of the body is kept separate from the outside environment. There has been evidence that diet can impact GPCR activity, especially with this GPR43 right here. So expression of this GPR43 has been reduced in intestinal mucosa of mice that are eating a diet high in fats and sugars. So it is possible that the diet is shifting the gut microbiome composition of the captive red wolf, thus changing the concentrations of these SCFAs. And while these SCFAs have a lot of beneficial impacts on immune function and intestinal barrier integrity, there could also be negative impacts like inflammation from having sustained elevated levels of SCFAs. It is possible that there needs to be this optimal level of each SCFA for these beneficial impacts. There is evidence that too much or too little of SCFAs can lead to obesity or other metabolic syndromes in humans and mice. And so besides the fact that the red wolf continues to largely exist in captivity, the opportunity for reintroduction is quite possible for the red wolf. Therefore, we need every red wolf in the SSP population to be healthy in order to facilitate a successful reintroduction. Recently, there was a paper put out by Yao et al. That introduced the idea that the gut microbiome takes longer to acclimate to the wild environment after a reintroduction than previously thought. This study looked at captive-born pandas released into the wild in China. And so they had these 3 different phases in their study. So they have when the panda cubs are just in captivity, when they're in the semi-wild phase, and then when they're wild on their own in their natural environment. Authors saw that gut bacteria composition and function dramatically shifted during this process, with the captive wolf gut being dominated by this Enterobacteriaceae, which is this light blue color here, compared to the wild pandas whose gut was dominated by this Clostridiaceae, which is the green, and the Pseudomonaceae, which is the red. So these differences in the gut bacteria community composition can lead to poor energy absorption, indicating that the giant panda and other species may need a longer time in this semi-wild phase to allow for the gut microbiome to acquire more wild bacteria before being released fully into the wild. This is a great example of why we need to strive to keep the gut microbiome community as wild as possible in the captive red wolf. The goal is to eventually reintroduce these animals into their historic range. And we want to retain the natural red wolf gut microbiome in order to protect these individuals from opportunistic pathogens and to ensure proper nutrient transformation, vitamin production, and communication between the GI bacteria and the immune system. So to help address this link between diet, GI health, and gut bacteria further, future research should focus on collecting more wild fecal samples so we can build a better profile of what the wild red wolf gut microbiome looks like. And then we could think about altering daily diet, so including whole meat and kibble for a daily diet, while simultaneously monitoring the gut microbiome of those individuals to watch for community shifts more towards the wild microbiome profile, which is what we want. We could also go back and assess total abundance of these important bacteria phyla, like the firmicutes, fusobacteria, and the bacteroides. Because like I said, in my study, I just looked at relative abundance, which is proportion. So there are ways that you can go look at total abundance that may give us a more detailed picture of these fluctuations that are occurring within the gut. And lastly, if the captive wolves were to be reintroduced, then it may be necessary for the captive red wolf gut microbiome to closely resemble the wild red wolf gut microbiome prior to release. And so in conclusion, our strongest finding is that the gut microbiome is in fact influenced by diet in the red wolf. We found that wolves consuming a wild diet type had lower bacterial richness than their captive counterparts, suggesting that captivity may be introducing these novel bacteria into the captive red wolf gut microbiome, altering it from its wild state. We found that the kibble diet had a relative increase in firmicutes, and the whole meat diet had a relative increase in fusobacteria. And it is possible that these captive red wolves are acquiring these non-native bacterial taxa because of their functional role in digesting these captive diets. However, these bacteria may not be beneficial for the red wolf. There are less than 300 red wolves left in the world, with 95% of individuals living in captivity. While this captive population is absolutely necessary for the continued existence of this species, the captive environment does bring manipulated diets. These factors, along with others, can cause shifts in the gut microbiome, negatively impacting the GI health of the red wolf. It is essential for the captive population to be healthy to facilitate successful reintroductions of these animals into their natural landscape in the future. And with that, I would just like to thank my committee for being my guiding light throughout this entire process. I would like to thank all of the facilities that donated their fecal samples and time to me, including Wolf Conservation Center. And I would also like to thank the U.S. Fish and Wildlife Red Wolf Recovery Team for letting me bug them for a week and hang out with them. I would like to thank Point Defiant Zoo and Aquarium's Dr. Holly Reed Conservation Fund for funding my research. And last but not least, I would like to thank all of you for showing up tonight and listening. I really appreciate it, and I really hope that you enjoyed my talk. And with that, I will take any questions.
Morgan, thank you so much. So just for those who might have joined us after the introduction, we are here now with Morgan Bragg, who just finished her talk on the relationship between the gut biome and gastrointestinal health in captive red wolves. And again, if you do have questions, please type them into the Q&A box in your control panel, and we'll get to as many as possible. So we have definitely the questions are coming in. Let's start with this one. I work at a wolf sanctuary in New Mexico. In the past, we've been concerned about feeding out potentially slightly rotten meat. I know that in the wild, they will eat some rotten meat. Is that important to maintaining a good healthy biome? Should they be getting a certain amount of rotten meat in their diet, or should they be getting meat that has zero evidence of rot/bacteria?
Yeah, so this is a good question. And so yes, you know, maybe in the wild they may be scavenging on carcasses or something like that. But I think with the red wolf, I think we want to think about safety first, especially because of how endangered these species are. I think that with the red wolf, we should just try to keep it as safe as possible with the meat. And so I know that it can be challenging for facilities, and so that's why, you know, we're trying to figure out the best diet type for them. But
that makes sense. And there's a question wondering, how long did it take, if you know this, domestic dogs to adapt to a starchy diet?
mm-hmm. Yeah, so if I remember correctly from reading literature, I think the event of domestication happened around 10,000 years ago from the wolf. And I know obviously that back then, you know, these domestic dogs weren't eating this like dry dog kibble that we think of feeding our dogs, but there was a lot of agriculture process and development going on at that time. And so there is theories that this domestication process happened because these wolves were kind of attracted to this waste from their agriculture development. And so that's how that relationship started. So I don't know exactly like how long, but that is the information that I do know about that.
And just kind of a follow-up, would we realistically expect them to adapt to digest starch as well? And that's referring to red wolves?
So my opinion is that I would not expect them to be able to digest starch as well as the domestic dog, just based off some of the things that I showed you guys today in literature. It just looks like that's where that evolutionary split really happened, was between the dog and the wolf, was the ability to digest starch.
Got it. Okay, this person's asking, do you think any, and I believe you covered this a bit, any of this would also have to do with stress levels as well as poor food quality, and how as individuals we can positively affect captive red wolves being fed wild food as they would in the wild?
Okay, so I'm gonna address the first part first.
Great.
With the stress. So I actually, for part of my PhD, I am looking at how this fecal metabolite called fecal glucocorticoid metabolites, which has been associated with stress before. It's not a stress hormone, but it has been associated with stress. So I am assessing how levels of fecal glucocorticoid metabolites relate to gastrointestinal health. So I will have some more answers for you in probably a couple years, but not yet.
All right, we'll follow up then.
But it is, let me just say though, it is possible for stress to impact the gut microbiome.
All right. Is it possible to use probiotics or fecal implants to normalize the red wolf gut bacteria to better resemble wild red wolves?
Yeah, so there has been some success in humans with fecal microbiota transplants and things like that, but I just do not think that we are at the level right now to be prescribing probiotics or doing any of these fecal microbiota transplants. I think we have a lot to learn before we can get to that level first. When you do give probiotics, it has to be very specific. And since we just don't have a lot of that information yet, it's hard to design this very specific probiotic for them.
All right. A lot of people just saying great job to you.
Thank you guys.
Yes. In providing a diet of whole meat, will organic meats offer a reasonable substitute for natural prey?
I honestly don't have a sound answer on that. I could hypothesize. I mean, I think that whole meat in general would be more beneficial than the kibble. And I haven't seen any literature looking at like organic versus not organic or anything like that. So sorry, I can't give too much information on that one.
That's okay. Here's an interesting question. The bacterial species that are more prevalent are also present in soil. And this poses a question: does it make sense to import soil from a reintroduction area to the prime gut, or to prime the gut, excuse me?
Yeah, you know, I've actually thought about this idea before because I've read some literature, especially with like ungulates and species that are browsers that do pick up a lot of soil when they're eating. When they are transferred to these different facilities, they have seen these issues with gut microbiomes. And I have seen where they've imported soil from like the old facility to the new facility to try and help with that. So, I mean, it could definitely be a possibility. I have not looked into it. I haven't seen anyone look into it specifically with the red wolf. It definitely could be a possibility though, because I mean, they are eating the food off the ground.
That is interesting.
Yeah, very interesting.
Yeah, and this one is a poop question. Is it difficult to get, yeah, I bet. Is it difficult to get an accurate reading of the microbiome from found scat samples, i.e. scat sampling in areas with wild red wolves? And how long does a scat sample remain an accurate representation of the gut microbiome before it's too old or dry?
Right, yeah, absolutely. That's a great question. So with my captive facilities, it's a bit easier to manage that because I did ask for everyone to collect the fecals within 1 hour of defecation to try and avoid this. And once it was collected, it was supposed to be immediately put into a freezer so we could preserve that bacterial DNA. I have, when I went to Alligator River, so we set the trap in the night, and then in the morning was when we found that those 2 fecals. So it did sit overnight, but with wild animals, I mean, I kind of had to take what I could get with that. It was very opportunistic, and I didn't want to not use it, basically. I thought it was really valuable information. And I have seen studies that are looking more into how long fecals sit out and if they're still viable. And I was actually just reading something today, and it was showing that, I mean, up to a certain point in time, I can't remember the exact time, but the fecals didn't really show any significant differences in the bacteria that they were, that the evidence that they were providing of the bacteria present there. So, of course, we want to try and get the fecals into the freezer as fast as possible, but sometimes it's just not possible in the wild.
Yeah, or even in captivity.
Yeah, no, in captivity too.
Yeah.
Yeah, it's really hard. I know for you guys, it's hard for you to just sit there and watch a wolf until they poop because you got a million of other things going on. So it is definitely a tricky thing.
Sounds like a good job for an intern. Just kidding. So some 2 related questions. One is, would there be negative impacts on the captive red wolves if they were fed factory farmed animals like beef or pork? And a separate question asks about pork specifically, and if that's has any negative impact on their biome or health.
Yeah, again, I'm sorry I can't be too detailed with this answer because I don't know. I haven't seen a lot of literature looking at feeding them like beef, cattle versus pork or anything like that. I do know, like I said, with that mixed diet, those logs are typically made up of horse meat. But unfortunately, I'm sorry, I just don't have a lot of information on that.
Don't worry about it. All right, this is a good one. It states, I would think more bacteria is better. Is that true? I'm surprised that captive wolves have more bacteria than wild wolves, if that's correct.
Yeah, so my study was actually kind of interesting because some other literature that I had been reading, it was reverse, where the wild individuals had a higher number of bacteria present compared to the captive wolves. So it was pretty interesting to find that. But again, I think that it's more about just having these like good levels, not like too much or too little. So I don't know if saying that having a lot of bacteria is necessarily beneficial. But I think it's all just about having this good balance in your gut, which is tricky.
For sure. This is an interesting question. If a wolf grows up eating kibble, but then is fed a raw diet, will their gut reset?
Oh, interesting. So I'm assuming reset back to like the kibble gut microbiome. Interesting. So I mean, I guess if your wolf was being fed kibble and then they shifted to a meat diet, then their gut would also shift because they are being fed a diet. And if they are continuing to be fed that meat diet, I wouldn't really expect it to be going back too much to what the kibble diet looked like unless their diet was changed as well.
Okay, and then it follows up. If they grow up on a raw diet, does it create a healthy environment that's still present if they're then switched over to kibble or mix? So I guess, is there something about that initial diet to create that environment inside the gut? Is that what will generally stick around, or does it really fluctuate with their diet at the moment?
So the core microbiome that, so everyone actually is born without anything, and then either it's given to you via breast milk or the way that you were birthed. So people who are vaginally birthed tend to have higher number of bacteria present in their gut when they're babies. So a lot of it has to do with their environment, like when they're born. And then, yes, diet can be impacting it as they go on throughout their life. So it really is important to have a good core microbiome when you're born. So then you are kind of basically like set for life, even if you did have these kind of fluctuations, versus if you were born and you didn't really have a solid core microbiome, then any of these fluctuations would probably have a even larger impact on you. You wouldn't be able to kind of be as resilient.
Okay. This is a kibble question. Are some kibbles more wild than others? For example, premium kibbles that are advertised as being more similar to a wild diet.
I mean, some kibbles, they all have different percentages of whether it be protein, carbohydrates, fats, things like that. So I know that there are some dog foods that do like broadcast themselves as being more like towards a wolf thing. And I know the wolves in my study, their kibble had to be between 20 to 25% protein to be fed. But yeah, so I guess there are like some that maybe broadcast themselves as that, but I don't know off the top of my head.
You don't know all of your dog foods, come on.
No, I'm sorry.
A lot more comments just saying thank you and congrats, and really wishing you the best, Morgan. So I'm just gonna
thank you
skim
guys
through
so
these.
much.
Here's something. The most recent study I've seen on wolves and dogs regarding the relationship on the evolutionary tree is a paper that came out about 4 years ago concluding that wolves and dogs actually split off from a common ancestor. Oh gosh, this is gonna, I don't even know how to say it. Taimyr Peninsula of Siberia over 34,000 years ago. That is, dogs are not a descendant of wolves, but they share a common ancestor. I'm wondering where the concept of agriculture fits in with this. I don't think of Siberia as being a place where there was a lot of agriculture taking place nearly 35,000 years ago. So this is puzzling. Any thoughts on this?
Yeah, that's a great question. Yeah, that's a great question. I mean, yeah, there may not have been a lot of agriculture development because the temperature, the climate is quite cold. But I don't know, maybe just the fact that, I mean, humans just have waste where we're living, and this waste could have been attracting these wolves, and humans could have kind of got this, could have been interested in these wolves and could have taken these wolves in that maybe were less aggressive towards them or anything like that, and maybe domestication could have spiraled out of that relationship there. But that's a really interesting point of view to think about.
No, definitely. And that's just comparing wolves to wolves. Have you compared the red wolf gut biome with the gray wolf gut biome?
Yeah, so there's only one study that I really saw that looked at wild gray wolf microbiomes. It was out of China. And the red wolf, interestingly enough, it kind of, I had relative abundance plots, and I had the domestic dog here, I had the red wolf in the middle, and I had the gray wolf on the end. And it was kind of interesting because the red wolf looked like almost like a middle ground between the domestic dog and the wild gray wolf. So it was kind of interesting. And again, the red wolves that I use, right, they're mostly captive. But I thought it was quite interesting to see that it really kind of lied in the middle between the domestic dog and the wild gray wolf.
Oh, sorry, I was muted.
It's okay.
My bad. In the cases of inflammatory bowel disease in wolves, are they returned to the wild type diet? If so, does this resolve the IBD and revert the gut biome to something similar to red wolves in the wild?
Well, we actually don't really have a great grasp of what the wild red wolf gut microbiome profile looks like. Mine was the first study to do that, and I only had 2 individuals, which is great, but I would like to have more, like I said, to get the better profile built. And so, like I said before, I did have those 2 individuals in my study that were diagnosed with IBD, and they were put on that whole meat diet. I know that they, well, not specifically with those females, but I know just in general that they have seen like better fecal consistencies with like altering the diet. But I know that these wolves also take medication, so whether it be steroids or other things to try and manage for this IBD. So I think that there's just there's a lot of things going on in those animals. So I guess don't exactly know where their gut microbiome ends up, even if they are being fed this whole meat diet.
Got it. And I guess the last question, again, it follows a congratulatory note and asks, what are your plans for the future? Looking forward to more work coming from you. So what are your plans?
Oh, thank you so much. So my plans right now, I actually just finished the first semester of my PhD. So very exciting. And I'll probably be here for the next 3 years or so. But I'm going to be looking at, like I said, how stress is impacting gut health in the red wolf. And I'm also going to be looking at gut microbiome composition and how it's related to gastrointestinal health in the maned wolf, which is another canid species that in captivity suffers pretty badly from inflammatory bowel disease. So I'm really excited to keep going down this path. I love looking at gut microbiomes and I love microbial ecology and trying to understand these complex interactions between these bacteria and the environment. And then once I get my PhD, I'm hoping to work for a federal agency somewhere. So whether it be US Fish and Wildlife, USGS, Park Service. But yeah, thank you for all the congratulatory things though.
No, seriously, lots of well-wishing. Really awesome. So, you know, Morgan, we really appreciate you really talking about this really interesting topic. Is there anything else you want to add before we wrap things up?
No, I just would like to extend a huge thank you to you guys at New York Wolf Conservation Center for not only participating in my study, but for asking me to do this. I was so excited to share my work with you guys. And just a huge thank you to my committee, all the facilities that participated, and everyone for showing up. I really appreciate it.
Oh gosh, it was easy for us. We'll thank the wolves for on your behalf.
Yeah.
They did all the work, right?
Yes.
So, and everyone else, we really appreciate you being here too. And if you would like to learn more about the Wolf Conservation Center, our scientific webinar series, or the 42 wolves who currently call the Wolf Conservation Center home, please visit our website at www.nywolf.org. Also, our next webinar is on June 11th, and it's part of a special crime series we'll be offering. It's called Wild Crimes, the exploitation of our world's most vulnerable animals. And registration is open. You can sign up via our website. So just go under special events. And in the meantime, again, Morgan, thanks again for really just kind of opening this whole new gut world to us. And again, everyone else, we hope to see you on June 11th. So have a wonderful night, everyone.
Thank you.